All right. I just wanted to make a short announcement, and that is that we're accepting letters of intent for the Alfie Milne Young Investigator Award for researching CLAs. So this is a one-year award for $25,000. And I posted the link in the chat for the um, announcement. And you can also find it on the LMI website as well as the LGDA website. So it's my great pleasure to introduce our distinguished speaker today, Charles Griffin McDaniel. Griffin earned a BS in chemistry and a BA in neuroscience from Duke University in 2021. He's currently pursuing an MD PhD in the lab of Dr. Tim LaCroix at Cincinnati Children's Hospital. And in Dr. LaCroix's lab, Griffin is utilizing in vitro and in vivo models to investigate the pathogenesis of KLA. He's been an exceptionally productive student, publishing several articles, including an outstanding review on KLA and a description of a KLA mouse model. Griffin has presented his exciting work at numerous international conferences, including the ISFA conference, the International Vascular Anomalies Conference in Berlin, and also the LMI, LGDA, and LGDA Europe International Conference on CLAs. He's also received several awards, such as the Power of One Fellowship, and was one of the 2024 recipients of the Alfie Milne Young Investigator Research Award on CLAs. The title of his presentation today is Mouse Models of KLA. Please join me in welcoming Griffin.
All right. Well, thanks, Mike, for the introduction. So my name's Griffin McDaniel. I'm an MD PhD student in Tim LaCroix's lab at Cincinnati Children's Hospital. And today I'll be going through some of our recent work in developing novel mouse models for Kaposiform Lymphangiomatosis, or KLA. Kaposiform Lymphangiomatosis, or KLA, is a pediatric and adult disease with very low survival. So survival is about 34% overall. Patients with KLA experience bloody fluid that builds up in the chest. They can have low platelets. They can have significant problems with blood clotting. The disease is characterized by multifocal lesions of Kaposiform or spindled lymphatic endothelial cells that accompany malformed lymphatic channels. These lesions can present anywhere in the body, but most typically show up in the lungs, spleen, and bone. They can also rarely occur in the liver. And a somatic activating NRAS Q61R gene mutation has been associated with KLA. Now, this is a somatic mutation, which means it occurs sometime post-zygotically, so it's not an inherited mutation. And we don't know when it occurs. It could occur sometime during development or sometime after birth, but we do know that it is a somatic mutation with KLA. We'll go through some pathology of KLA. These are slides from lung sections of a lung lesion from a patient with KLA. First is an H&E, a hematoxylin and eosin stain, which just shows the typical architecture and layout of the tissue. So this lesion is very abnormal to see in the lungs. We see spindled cells. We don't see any typical lung tissue in this section. We can immunostain for PROX1. PROX1 here is a transcription factor that is a marker of all of the lymphatic endothelial cells. So in this slide, it's showing up as all the brown color. And we see that these lesions are composed of a large proportion of PROX1 positive cells, which is telling us that they are lymphatic endothelial cells predominantly making up these lesions. Here's another H&E. The bright red color here are actually red blood cells. So this is showing us that the involvement of the blood system in these lesions. And lastly, here's a chest CT, some imaging of a patient with KLA. Typically, the lungs appear as black in a CT because they're full of air and they're very low density. Now, in this patient, there's extensive fluid buildup and effusions in the lungs, and that's all the dark gray color kind of in the back part of the lungs in this patient. And this is highlighting the extent of the effusions and really the severity of the disease. Now, because these patients are so sick, obtaining biopsies can be quite risky. It's not always safe or not always feasible for patients with KLA. And so non-invasive biomarkers have real utility in helping clinicians come up with a diagnosis. So in 2017, Lacrade all published that angiopoietin-2 levels are highly elevated in the blood of patients with KLA and KHE. This is really useful for distinguishing KLA from some of the other complex lymphatic anomalies, such as generalized lymphatic anomaly or GLA, in which case angiopoietin-2 levels are actually quite low. Now, angiopoietin-2 levels are also elevated in KHE or Kaposiform Lymphangiomatosis, which is another vascular anomaly. However, it's not a complex lymphatic anomaly, and the clinical presentation can help distinguish KLA from KHE. This test has been developed into a clinical test that's available here at Cincinnati Children's for samples from other institutions as well. The NRAS Q61R activating gene mutation that's been identified with KLA patients is present in the initial description that discovered this mutation in the vast majority of patients with KLA. So it was found in lesions of 10 of 11 KLA patients. NRAS is a proto-oncogene in the RAS gene family that encodes for a small GTPase involved in cell proliferation. And importantly, this gene mutation has been found in many cancers, including about 20% of melanomas, thyroid cancers, and other tumors. And it results in increased activation of MAP kinase and PI3K signaling pathways that causes unchecked cell proliferation in tumor models. And now I'll give a little overview of how NRAS is functioning. NRAS is this is a depiction of typical NRAS functioning, so not a mutated form of NRAS. And in the blue on your screen is the cytosol, or part of a cell, and then in the white is the extracellular space. And so typically when NRAS is off, there's a receptor tyrosine kinase, which is not bound to anything, and NRAS in the off state is bound to a molecule called GDP. When a growth factor binds to the receptor tyrosine kinase, what happens is that activates a protein called a GEF, which exchanges the GDP molecule on NRAS with a GTP. When NRAS is bound to GTP, it's in the on state. When NRAS is on, it can activate PI3K signaling and RAF-MEK-ERK signaling, resulting in cell proliferation and migration. Normally, this is tightly regulated. So when a growth factor binds, NRAS will turn on, and when the growth factor is gone, NRAS will be off. However, with the NRAS Q61R mutation, NRAS is locked in the on position, bound to GTP. So it's always active, always signaling through PI3K and RAF-MEK-ERK signaling pathways, even in the absence of growth factor stimulation. It's impossible to turn it off when it's mutated in this way. So we know from the pathology of patients with KLA that they have spindled endothelial cells. We also know from magnetic resonance lymphangiography imaging that they have abnormal lymphatics. So in the center of your screen is a picture of a magnetic resonance lymphangiography from a patient with KLA, just demonstrating the extent of the abnormal lymphatics and these enlarged lymphatics throughout the chest and abdomen. We also know that angiopoietin-2 levels are highly elevated in patients with KLA, and this is a biomarker that can be used to help clinicians diagnose patients, and further, it can be used to monitor response to treatment. And we also know that this NRAS Q61R mutation has been associated with KLA. And the major questions we're addressing are, does the NRAS Q61R mutation cause the spindling of the endothelial cells and the development of these abnormal lymphatics? And do these cells produce elevated angiopoietin-2 levels, as is seen in patients with KLA? To study this, we've generated an in vitro model in the lab using doxycycline-inducible NRAS Q61R and NRAS wild-type lentiviral vectors. Now, these lentiviral vectors have NRAS Q61R gene, or in our controls, the NRAS wild-type gene, downstream of a TET-responsive promoter. So when we add doxycycline, which is an antibiotic, to the media, it turns on expression of NRAS Q61R, and in our controls, NRAS wild-type. We've transduced these cells into, or transduced these constructs into human endothelial cells by lentiviral transduction. We've selected for the cells that uptook these lentiviral vectors with pure myosin, and then we've expanded these cells in culture. And so we can use these cells when we add doxycycline to the media to turn on expression of NRAS Q61R in these endothelial cells to study cell shape and signaling, migration and proliferation, and Ang2 production in the presence of this gene mutation. The most striking feature of these cells when you just look at them under the microscope is our NRAS Q61R endothelial cells develop a spindled morphology. So the image on the left is a cells expressing NRAS wild type, and these were our controls that adopt a typical cobblestone-like appearance on the cell cultured plate. However, NRAS Q61R cells develop this elongated and spindled morphology. Dermot Fox, who's an undergraduate in the lab, wanted to look at this over a course of time. And so he did some time-lapse microscopy of these live cells. And when the movie starts is when doxycycline is added to the media. And what you'll notice is when dox is added, the cells start to develop this elongated and spindled morphology that becomes more pronounced with time. These NRAS Q61R human endothelial cells also display increased migration and proliferation. There's some data showing the extent to which these NRAS Q61R cells proliferate more than the controls. The NRAS Q61R cells are in red and the controls are the other colors far below. We've also shown that these cells have hyperactive MAP kinase signaling, increased angiopoietin-2 production, and develop abnormal vessels in xenograft models. And while this is really useful, we have great temporal control over when we turn on and off expression of NRAS Q61R and ease of experiments. There are some limitations to these in vitro models. First, the cells are in a 2D monolayer, whereas a vessel structure obviously has 3 dimensions. And so we may not be fully recapitulating the vessel structure characteristics in this 2D monolayer. The cells in the dish, we have a single cell type, whereas a lesion from a patient with KLA will have multiple cell types and is much more complex than we may be getting in our cell culture dishes. And lastly, our experiments typically have shorter time courses with this in vitro model on the scale of days to weeks, whereas patients with KLA might experience disease development and progression for months or years. And so we may not be fully mimicking the time course of the disease with our in vitro models. And so there's also a need to develop in vivo models that we can use to study KLA. Our most recent objectives have been to generate genetic mouse models with targeted NRAS Q61R expression, and we want to use these models to study the disease-causing pathways in KLA, and when in development the NRAS mutation occurs to cause disease. We've chosen to do this with a mouse model, and there are numerous advantages to using mouse models. It is an in vivo mammalian model system, and so we have full organ systems in the mice. We can study disease development and progression over time. And these mice more closely resemble the cellular complexity of disease tissues because we have multiple cell types that are present in the mouse, whereas in our in vitro models, we have those single cell types in a monolayer in a dish. Additionally, with mouse models, we have available to us a huge toolkit of genetic tools so we can introduce gene mutations and cells in which we want to study. We've done this with a pan-endothelial NRAS Q61R mouse model approach. And the way this works is we first took a lock-stop-locks NRAS Q61R mouse. This was initially developed by Norman Sharpless to model melanoma, actually. So in this mouse, there's an NRAS Q61R gene downstream of stop codon. So typically, the NRAS Q61R gene is not being expressed because there's this stop codon that sits right in front of it. We have crossed it to a cadherin 5 Cre ERT2 mouse. This mouse expresses Cre ERT2 and all the cells that also express cadherin 5, which are all the endothelial cells of the mouse. So we get expression of Cre ERT2 in all the endothelial cells of the mouse. Now, this Cre ERT2 is in its resting state inactive, and when we add tamoxifen to the system, which is just a drug, Cre recombinase becomes active, and Cre recombinase performs its function, which is to cut out the stop codon in front of NRAS Q61R. So in our pups that have the alleles for both cadherin 5 Cre ERT2 and lock-stop-locks NRAS Q61R, when we add tamoxifen to the system, we cut out the stop codon in front of NRAS Q61R, and we express NRAS Q61R in all the endothelial cells of the mouse. As I mentioned earlier, to do this, we have to add tamoxifen to the system. The way we do that is we give tamoxifen orally at postnatal days 1, 2, and 3. We just pipette it into the mouth of the pups. We didn't observe any mortality in the mice, and so we conducted all of our analyses at an 8-week time point. At 8 weeks, we collected plasma, which we measured angiopoietin-2 levels in the plasma by multiplex. Angiopoietin-2 levels were significantly elevated in our NRAS mutant mice compared to our controls. And this recapitulates what is seen in patients in whom angiopoietin-2 levels are highly elevated. We also ran complete blood counts on the blood from these mice. We found that the platelet counts were significantly reduced in our NRAS mutant mice. So this is recapitulating the low platelets that are seen in some patients with KLA. And then lastly, the spleen to body weight ratio was significantly elevated in our NRAS mutant mice. We corrected the body weight just to account for any differences in the overall body weights of the mice. So we're sure that the increase in the spleen size is an effect of the disease and not just an effect of all the mice having bigger spleens that are our NRAS mutants. Splenomegaly or enlarged spleens are also seen in some patients with KLA. Not only are the spleens enlarged, but they also have some discrete and focal areas of lesions or diseased or damaged tissues that you can actually see with your eyes visible on the surface of the spleen. And those are pointed out with the white arrows. So put together, these NRAS mutant mice are mimicking a number of the clinical characteristics of patients with KLA. We wanted to look at some histology to figure out what exactly is happening within these splenic lesions. First, we took tissue sections and we stained with hematoxylin and eosin just to look at the tissue architecture. And we see very irregular structures, and this is not at all what normal spleen histology looks like. So there's total disruption of the typical splenic architecture. There's expansion of spindled cells within these lesions. And furthermore, there are all these red blood cells that we can see in the H&E stain present in these lesions. So this is recapitulating some of the pathology that's seen in KLA patients. We immunostain for CD31, which is a marker of endothelial cells, to try to figure out which cell type is making up these lesions. CD31 here is going to show up as black, and so we see that these lesions are composed of a very significant population of endothelial cells. We've called them hypervascular. We've also stained for LIVE1. Again, this will show up as black. LIVE1 is a marker of lymphatic endothelial cells. And what this stain shows us is that inside of these lesions, there are enlarged and dilated lymphatic vessels. So to summarize our pan-endothelial NRAS Q61R mouse model, our NRAS mutant mice are mimicking some of the clinical characteristics of KLA, including elevated blood angiopoietin-2 levels, low platelets, and enlarged spleens with hypervascular splenic lesions. However, cadherin 5 Cre-ERT2 is a pan-endothelial driver of Cre recombinase. So we're expressing NRAS Q61R in all of the endothelial cells of the mouse. And KLA is a complex lymphatic anomaly. And so our next objective was to study what happens when we express NRAS Q61R specifically in the lymphatic endothelium. We've taken a similar genetic mouse model approach to do this. For this, we've taken the same lock-stop-locks NRAS Q61R mouse, in which case there's a stop codon in front of the NRAS Q61R gene. We've crossed it to a LIVE1 Cre mouse. So this mouse is different than our Cre in the previous experiments for two main reasons. First is Cre recombinase is downstream of the LIVE1 promoter. So we're going to express Cre recombinase in all the cells that are also expressing LIVE1. Additionally, this Cre recombinase is constitutively active. And so whenever LIVE1 turns on, we get expression of Cre recombinase, and it's going to immediately function. We don't need to add tamoxifen to the system. And we don't have temporal control over when we turn on expression of the mutation. So when we do this breeding, the pups that have the genes for LIVE1 Cre, as well as lock-stop-locks NRAS Q61R, as soon as LIVE1 turns on, Cre recombinase is present in the cells. It cuts out the stop codon in front of NRAS Q61R, and we get expression of NRAS Q61R in all of the LIVE1 expressing cells. These mice don't actually survive after birth, and so we've had to conduct all of our analyses at prenatal or embryonic time points. First, I'll show you some example images of our NRAS mutant embryos. And these are at E15.5, which is about 2/3 of the way through gestation. First of our controls look as typical mouse embryos at this time point. There's regular vascular patterning throughout these embryos, as well as the abdominal organs appear normal. Our NRAS mutant embryos have a number of striking differences from our controls. First, there's the buildup of fluid or edema directly under the skin throughout these embryos, and this is very severe. We see abnormal spots of hemorrhage or blood-filled vessels throughout the skin of the embryos that we don't see in our controls. These are highlighted by the red arrows. And lastly, it appears just grossly looking at the embryos from the outside that there are some differences in the abdominal organs. So we wanted to assess specifically what is happening inside of these embryos. And because we're interested in the lymphatic system, we first wanted to look at the jugular lymph sacs. The jugular lymph sacs are in the neck regions of the embryos, and they're part of the initial development of the lymphatic system. What happens are a subset of cells in the cardinal vein start to express lymphatic markers. They migrate from the cardinal vein and actually form these initial jugular lymph sacs, which we're looking at and which we're studying here. Here are the jugular lymph sacs in our control embryos. As I mentioned earlier, we are in the neck region of these embryos. That's where we're finding these structures. And to orient you, first in our control H&E, hematoxylin and eosin stain, just to show normal tissue architecture. The carotid artery is in the bottom left in red. Just above that is the jugular vein, which is in blue. And above that is that large dilated structure, which we're calling the lymph sac, and has a LS sitting in the middle of it, standing for lymph sac. We know these are indeed the lymph sacs because when we immunostain for LIV1, which is a marker of lymphatic endothelial cells, we see that the lymph sac is lined by LIV1 positive cells, whereas the jugular vein and the carotid artery are not. And in our controls, when we stain for NRAS Q61R, we don't see expression of the mutant protein, which is what we'd expect in our controls. Now, in our NRAS mutant embryos, the jugular lymph sacs are quite different. So first, they no longer have this normal structure that our control embryos have. They have these dysmorphic borders and these protrusions of cellular masses within the jugular lymph sacs. We know these abnormal structures are the jugular lymph sacs because when we stain for LIV1, we see that these dysmorphic borders of the lymph sacs and these abnormal protrusions of cells are LIV1 positive. And when we stain for NRAS Q61R, we see expression of NRAS Q61R in these abnormal appearing structures, which is suggesting to us that NRAS Q61R is driving this phenotype in the embryos. As I mentioned earlier, the abdominal organs of these embryos appeared quite different in our NRAS mutant embryos. And so we wanted to look more closely at what was going on in the abdominal cavity. And so we dissected out the abdominal organs, and we're mostly interested in looking at the liver. The liver has very strong expression of LIV1 throughout the liver sinusoids. Now, while these are not exactly lymphatics, we still think it's useful to study the effect of the NRAS mutation in endothelial cell biology more broadly. So first, these are just gross pictures of the whole liver under a dissecting microscope of our controls. This is very normal appearing for an embryonic liver at E14.5. And then of our NRAS mutant embryos, here's another picture under the dissecting microscope. And they look quite different, very irregular vascular patterning that you can kind of see from the outside. And then furthermore, there are these abnormal pockets of blood pooling that we can see. We then wanted to look at this with microscopy, and so we did some three-dimensional confocal imaging, in which case we can actually see the vessel structures in 3 dimensions. Here's where we stained for LIV1 and then did our three-dimensional imaging. In our controls, we see very regular patterning of the sinusoids of the liver at E14.5. And then in our NRAS mutant embryos, they are LIV1 positive vessel patterning is quite different. So it's very irregular. And then additionally, these LIV1 positive vessels are enlarged and dilated compared to our controls. Then we looked at the livers through traditional two-dimensional section histology. Our H&Es and our controls show very normal liver architecture at E15.5. Staining for live 1 shows very robust expression of live 1 throughout the livers. And then staining for NRAS Q61R shows no expression of NRAS Q61R in our controls. And this is all what we'd expect to see. This is very normal of embryos at this time point. Now, in our NRAS mutant embryos, H&Es show these abnormal pockets that are blood-filled in these embryos. When we stain for live 1, which is in the black, we see that there's very irregular live 1 positive vessel structures, and these abnormal pockets that are present in the liver are lined by live 1 positive cells. And we also see in these diseased areas of the liver strong expression of NRAS Q61R, further suggesting to us that NRAS Q61R is causing these abnormal phenotypes. And lastly, we wanted to look at the lymphatic vessels in 3 dimensions. So we worked closely with Dr. Dellinger to develop this protocol and get it up and running in our lab. And effectively, what we've done here is we carefully dissect the back skin off of the embryo. So we peel the back skin away from the remainder of the embryo. We can stain for markers that we're interested in looking at. And then we can mount that on a slide and perform 3-dimensional confocal imaging. Here is an example image of a control embryo back skin, which we've stained for neuropilin 2, which marks the lymphatic vessels in the back skin. This control embryo shows very normal appearing lymphatic vessels. They're of normal diameter and normal branching and normal patterning. However, our NRAS mutant embryos show severe differences in the lymphatics of the back skin. Most notably, the vessel diameter in our NRAS mutant embryos is significantly enlarged compared to our controls. And there are fewer branch points, showing that there's this irregular patterning of the lymphatic vessels in the back skin in our NRAS mutant embryos, showing that NRAS Q61R expression in the lymphatic endothelial cells is disrupting lymphatic vessel development in these embryos. So just to summarize what we've talked about in these 2 new mouse models for KLA, the first was our pan-endothelial NRAS Q61R expression mouse model. This is our postnatal model where we give tamoxifen to the mice to turn on expression of NRAS Q61R. These mice mimic some of the clinical characteristics of KLA, including low platelets, elevated blood angiopoietin-2 levels. They have enlarged spleens with hypervascular splenic lesions. In our live 1-driven NRAS Q61R model, this is our prenatal model, which we conduct our analyses at embryonic time points. We see the development of cellular masses and dysmorphic jugular lymph sacs. They have very abnormal liver vasculature. They have these enlarged back skin lymphatic vessels. So put together, these models are mimicking some of the clinical characteristics of KLA, and we propose that they can be used to further study disease pathogenesis and screen treatments in the future. So thanks to the whole lab for all of their contributions to this project. Thanks to our collaborators here at Cincinnati Children's and at external institutions for their help. And lastly, we'd like to specifically thank our sources of funding who are letting us do this kind of work. We have funding from the Lymphatic Malformation Institute, from the Ferry family, and I'm fortunate to have funding from the Alfie Milne Young Investigator Award and the Power of One Fellowship from the Steele Prokopowicz family, and then others who donated. We're also supported by Dr. LaCroix's NIH funding as well. So thanks for attending, and I can take any questions.
Great presentation, Griffin. I'll get the discussion started, and if anyone else, when you have a question, you can either raise your hand and then unmute yourself, or just feel free to unmute yourself and chime in. The morphology of the NRAS mutant cells is really striking in vitro, and then also the spindling that's observed in vivo. I'm curious what your thoughts are as to what's driving this change in endothelial cell shape, and whether or not you think this could be driven by an EMT-like mechanism.
Yeah, that's a great question, and one of ongoing research. What I didn't include the data in this presentation, but what we've published previously is that this spindled morphology of the cells can be blocked by trametinib, which is a MEK inhibitor. So those data suggest to us that in fact the spindling of the cells is primarily driven through MAP kinase signaling. I presume there is very significant cytoskeletal rearrangements in these cells that causes the spindling morphology to occur. But we think it's primarily driven through MAP kinase signaling. Now, are our cells undergoing some sort of mesenchymal transition? We haven't really looked into that too much quite yet. I think it's a really interesting area to consider, though, and really consider the overall transcriptomic profiles of our cells and how they're behaving and how their fates or identities may be changing when we turn on expression of this mutation.
Go ahead.
Thank you very much, Griffin, for this presentation. I was just wondering about like you mentioned about the trametinib, and we know that it actually decreases the proliferation when you have like a lymphatic endothelial cells in culture. It doesn't kill actually the cells, just decreases the proliferation. What would you be expecting in this mouse models, like when you treat them with trametinib? Like what would happen in your, like, it's a broad question, but I'd like to get your opinion on this.
Yeah, that is a great question, and we haven't done the experiment, so I don't have the data to show you what would happen if we treated our mice with trametinib. But our in vitro data is suggesting that the spindling of the cells is in the angiopoietin-2 production by the cells can both be blocked by trametinib. So I would hypothesize if we treat our mice with trametinib, either our embryos or our adult mice, or more specifically our adult mice, we'd probably get some sort of reduction in the angiopoietin-2 levels in these mice. And then furthermore, I think we'd probably see some reversion of the pathology, but we haven't done the experiment, so we don't really have the data to show which phenotypes specifically are driven, which phenotypes can be blocked by trametinib and which phenotypes might not be able to be blocked by trametinib, but we might need other different targets to try to block those characteristics of the mouse models.
Brian?
I was trying to raise my hand and then I gave you a round of applause, but you deserve a round of applause anyway. So I'm following up on that question. Clinically, these patients sometimes can have a partial response to sirolimus, and that's often the first treatment they go on. And we know that from what you were showing us, NRAS can activate either the PI3K pathway and the RAS pathway. Do you think that you would see these are actually having the same morphologic changes, the same effects in your system, or do you think that the RAS pathway inhibition and the mTOR inhibition are actually benefiting the patients from different effects on the system?
I think there are different effects on the system. We've published some work with rapamycin or sirolimus, and our data is suggesting to us that the spindling of the cells and the angiopoietin-2 production are driven through MAP kinase signaling or trametinib. So what exactly is sirolimus doing when we inhibit that arm of the pathway? I don't really know why it's so useful clinically, because obviously it's useful it's the first line therapy for patients with KLA, but we don't see it decreasing Ang2 levels or reverting the spindled morphology in our in vitro models. So I think there's definitely separate mechanisms for how those drugs are benefiting patients, and we just don't know quite which phenotypes are driven through which pathway clinically.
Very interesting. And then it also raises the question of potential synergy if you use both of them. Great work.
Thank you.
I know that there's a lot of interest in mutation-specific inhibitors now, and I was just curious if you guys plan on testing any of these in your animal model.
That's another good question. There's so many inhibitors out there for mutation specific, and then there are obviously other inhibitors for all the pathways that I've highlighted that might be elevated. So it's an area of a lot of interest in the lab and in the field broadly to try to screen all of these drugs. And I think we haven't done it yet, but I think that's one of the big utilities of having these models is that we're going to be able to try all of these different drugs and see, which ones might be helpful and which ones might not. So it's an area of a lot of ongoing work.
Beth?
Hi, yeah, that was a really interesting talk. I was wondering if I don't know this literature very well, but do you know if, I think you said the angiopoietin marker is a target of the RAS MAPK pathway, or you suspect that it's acting through that pathway. So do you know then what it goes on to do? So what cells is it signaling to that's, and whether it's sort of autonomous or non-autonomous in terms of, affecting the lymphatic vessel phenotype?
That's a great question. So angiopoietin-2 is a potent molecule that's involved in angiogenic processes quite broadly. It's context dependent, and it's very, it seems to depend on, the particular system and the particular disease you're studying to figure out what exactly angiopoietin-2 is doing. People study it in cancer frequently. People study it in vessel remodeling and vessel development too. So what is it doing in our disease? We don't know yet. We're working on it. It's been a tricky question to really hone in on. So I can't give you any details about what exactly it's doing in the disease, but it's very feasible to think that it is playing a role in either the lymphatic vessel overgrowth, cell proliferation, or something like that, given its roles in other vascular diseases and tumor angiogenesis and things of that nature.
Thanks.
All right, if there are no additional questions, let's thank Griffin for giving an excellent presentation.